Hello, everybody. My name is Liang, and I'm a professor of computer science, biochemistry, and bioengineering. Unfortunately, I'm on a flight right now, so I record this video so that Ellen can play it. So my background is not in bioengineering, but in NLP or AI, natural language processing. But in the past 5 years or so, I've been kind of switching to using NLP or AI to understand and engineer biological languages, mostly RNA and DNAs. So in other words, my area is in the intersection of computer science or AI and linguistics and biology. And so I'll just talk very briefly about what I do and what I plan to do in collaboration with you guys. So we'll just be doing understanding and engineering, but more importantly on the engineering side. But understanding of RNA languages, we do both single sequence folding and homologous folding. And in both problems can be converted to natural language problems. So we adapt natural language algorithms to these RNA folding algorithms. And homologous folding is just, not just folding one sequence, but multiple sequences, multiple related genomes, for example, SARS-CoV-1, SARS-CoV-2 together, and not just folding them, but also aligning them all together. And you know that they need to fold into kind of similar shapes or similar structures. So this is linear triple fold, the first PNAS paper from College of Engineering at Oregon State. And we can use it to do COVID test kit design, which we'll talk about in the engineering part. The first part of engineering is the mRNA vaccine and the mRNA medicine. So here is a very exciting story, which I talked about a few weeks ago, where we reduced the mRNA design problem to a classical problem in natural language processing. Again, using AI methods, we were able to achieve more than 100-fold increase in antibody response in vivo than conventional codon optimization methods used by everybody in the industry, including Pfizer, BioNTech, Moderna, everybody. And this is the first Nature paper published from the OSU College of Engineering. And the Nature also published a news article. A famous reporter wrote a news article about my work. Remarkable AI tool designs mRNA vaccines that are more potent and stable. And I'm currently working with Gaurav Sahai from our College of Pharmacy on extending it to mRNA therapeutics, so not just vaccine, but also medicine, for example, to treat cystic fibrosis. But another direction of the engineering is to design diagnostics and sensors, like point of care devices to detect, say, RNA virus and DNA virus like SARS-CoV-2. And I'm working with Larry Chen and ECE to design molecular beacons, but it can also be used for PCR primers because it should target, in order to be highly sensitive, it should target highly accessible regions, meaning this kind of single-stranded regions of the target genome. Could be RNA, it could be DNA genome, so that the binding would be easier. So for example, you have a molecular beacon and you want the loop part to bind to the target genome, and you want target genome to be single-stranded or accessible, not the double-stranded. If you want to bind to the double-stranded regions, then you have to pay extra energy, right? And not only accessible, you also want it to be highly conserved so that your test kits will remain effective even with future variants, which you cannot predict. So our PNS paper, which I mentioned above, solves these two problems. And it stood the test of time with the arrival of Omicron, because our paper was published before Omicron, but when Omicron came in, our students looked back at the results, and Omicron did not change at any single position that we prescribed to be important or to be conserved. Whereas the standard test kits, PCR primers, from like China CDC, because they only use the Wuhan virus as a reference, and as a result, it got worse and worse, less and less effective as time goes on. Because with more variants, with more mutations, it's likely that your PCR regions are mutated and no longer conserved. So we are doing conserved and accessible regions for high sensitivity diagnostics tools for viruses. Thank you.